Great. Today, I'd like to introduce you to technology that allows you to profile tissue sections and especially the molecular profile. And I'd like to focus more on looking at some of the data and really what you can get out of it. So I'm just going to very quickly cover on how the technology actually works. So happy to follow up on that if there's more questions. Why do you want to do spatial analysis in the first place? I mean, you look at these two samples, and if you would molecularly profile them, you might use a bulk analysis. And then you might, for example, quantify the different types of cells, like tumor cells in purple and the immune cells in green. And then you might get a similar result. But as pathologists, you know that those two are pretty different from each other. So one is a hot tumor. You can see that the immune cells are able to penetrate into the tumor, and the other one is a cold tumor. The immune cells are locked out. So the prognosis for those two would be very different, although maybe when you just quantify the different cells or the profiles without spatial information, you might end up with similar results. At 10x, we see this as a great opportunity to help researchers to make new findings based on our tools. So we have a single cell platform and also this spatial transcript platform that I'm going to talk to you about today. And we see increasing numbers of publication. It was actually released in, the newest version was released in 2018. And already since then, we see really an increasing amount of publications for this. So I'll just quickly talk you through what this Visium spatial gene expression solution looks like, and then we'll dig into some data. So everything is based on this glass slide. It's the glass slide has the size of a normal microscope glass slide. You can see those 4 squares, and each of those you can place one tissue section. And then if you look in more detail, you can see that each of the squares is 6.5 times 6.5 millimeters, and then each of them is made up by 5,000 capture spots. So each of those allows you to capture RNA on the surface. So you section the tissue, you place it on top, and then you permeabilize the tissue section. And then the RNA can bind to the surface, to the capture probes there. And then you can prepare libraries and do next generation sequencing. And each of those spots has the probe there has a barcode, and the barcode is spatial. So that means that you have 5,000 different barcodes, and you know exactly where each barcode is located. So that allows us later on, when we've done the sequencing and got the molecular profile, to actually put the molecular profile back on top of an image, an H&E stained or an IF stained, you can choose that, image of your tissue section. And then you really can explore the data spatially. The advantage of this specific solutions are that you get a tissue-wide picture, so you cover really the tissue section. You don't, like in LCM or similar technologies earlier, you had to actually select the region you want to analyze. In this case, you get a profile from all the spots across the tissue section. So that really gives you a great overview. You can look at the biomarkers that you know. We'll look at a few later on, or you can discover new ones. And also as to a complexity to the degree that you like. So you can start from very few, add more and more, just all the way to the whole transcriptome. And this integrates easily with your existing laboratory equipment. You don't have to buy a specific instrument, but rather what you have on already, and what you normally do is gonna suffice. Mostly like a microscope, a cryostat, soon a microtome. And this really is a very fast workflow. Within one working day, you can go from a tissue section to a library, and then you do sequencing. You can do either whole transcriptome, but we also have targeted panels if you prefer that. I'll show an example of that in a second. The resolution of this is the spots that I mentioned have a diameter of 55 microns, and that means you get 1 to 10 cells per spot, depending on the tissue. You don't have to dissociate the tissue, but you actually just section it and place it on top. You can do co-detection by protein already now by doing an immunofluorescence stain with your favorite antibodies, and then you get the mRNA information from during the assay. So you can do combined analysis. And in the future, we will also release a product that has highly multiplexed protein detection features as well. You have streamlined data analysis that we provide free of charge, and the software for that, and then everything is kitted and ready to use. Just quickly about our panels. So we have 4 different panels so far, which allow you to really focus on the genes you're interested in, or you can do discovery on the entire transcriptome. And I'll focus a bit on the human pan-cancer panel, which has about 1,200 genes. And this is an example of the data. So we start from the left, you have the H&E image that you take during the workflow. Then in the next one, you can see all the combined, the genes and the counts actually from those 1,200 genes. So you can really see that we get some, we can really capture the heterogeneity in the sample. And then in the next one, you can see a subset of those, of that human pan-cancer panel, which is focused on breast cancer for this breast cancer sample. And then we have those 196 genes here, specifically in data analysis. You can just choose whatever you want to look at. And then in the last one, we can see an example of EACR-1, just to see where this is expressed. And then you can pick any gene you're interested in within the panel. This is compatible with many different tissues. We have a list online of the tissues we work with, most cancer types, and also all the way to mouse tissues and other tissues. And I also mentioned that we have the software. We have an analysis pipeline that processes the image and the sequencing data, and then you get an output that is very easy to work with, and then also one file that is called a loop file that you can open in your browser, in the loop browser on your on a desktop, so like a Mac or Windows, and just do the analysis yourself. Really, the exploration, at least the initial one. So what does this data type look like? So just really the potential of this solution comes from combining 2 data types. So you have the histology image on the one side, and then to the right, you can see the gene expression. And the gene expression, just quickly to explain, this is a clustering representation. So you have spots that you had originally on the tissue. You have them in the gene expression space, so to say. And when 2 spots are close to each other, the gene expression is similar. So you use all of the whole transcriptome data that we captured to do this, create these clouds. And when you have a cloud, these spots most likely are very similar in the gene expression. It might be similar cell type or a similar area within the tissue. So very likely similar morphology as well. And then you can combine that. And that's really unique for Visium, that you can go back and forth. You can look at the image, or you can start from the gene expression data. And now let's look at some data. So this was done by the University of Tokyo. They were actually able to pick up a mutation in this RNA measurement because the mutation is very close to the 3' end. So this is an interesting application where they looked at the GATA3 mutation and then wanted to see what does this mutation have, what's the effect of the mutation on the gene expression profile. So if we start from the left, again, we have the H&E image, and then we can see that they selected the cancer areas 1 to 3 here, and then also the microenvironment in different clusters. And then they looked here now at which of these spots do have the mutation and which of the spots don't have the mutation. So in green here, you can see all the spots that don't have the mutation. In red here, you can see all the spots that do have the mutation, and then you can see both combined in the third image. And really interesting, I encourage you to have a look at this preprint. What they found is that the mutation is really associated with an upregulated epithelial mesenchymal transition and angiogenesis, but also showed reduced estrogen response. So just to give you an idea of what you can do with this. Now I quickly want to just show you one example, which is a public data set on our website that you can download today and explore in Loop browser that you also can easily download free of charge. Just to mention about the interesting thing about this sample is probably that it is HER2 and ER positive, but PR negative. And we'll have a look at that in a second. So again, we start from the H&E image. And then we gave this to pathologists because we're not pathologists on our own. So we knew we're totally dependent on this knowledge who annotated this. And most interesting, we have invasive carcinoma in black and ductal carcinoma in situ in green. So the first thing that we can do is that we use something that is called k-means-2 clustering. And what that means is that we, as an output of Space Ranger, we have an algorithm that tells us about strong patterns within this data. And we asked, show me the two strongest. So I want to have two groups and show me those two groups that have the strongest difference. And what we see already now without using these annotations is that they already pretty nicely align with what we've seen in the morphology and from the pathologist. And then down here, you can see that cloud representation where you can see the outside clouds or the red ones, and then the orange one is the central one. But that's just the start. Now we can look at any markers we want to do. We can look at AbCam, HER2, ESR1, which we already knew it was positive for, and then also PGR, which we know it's negative for. But now we really just start to explore. So now what we can do is that we can actually select an area in the tissue, and we can do as many selections as we want, because what we've done is we captured all the data during the experiment. So you don't have to select a region and then, oh no, I selected the wrong region. I wanted to make it a little bit larger, a little bit smaller. All that you can do in data analysis. So no regrets there. So now we select that region. And now we want to look at, okay, I know this is ductal constant C2. And then on the top middle projection here, you can see, okay, they pretty nicely collected in the cloud. But what I could do now is, okay, now I use this information and I want to understand where the entire cloud is placed. So now I selected the entire cloud and looked at where are these spots located again in the tissue. And really nicely, you can see it's all the ductal constant C2 areas. And that's just based on the molecular profile. I didn't use the image at this point. And this is just the start. Now we can again look at a marker that might be specific, write any marker here that you're interested in and look at the different projections. But now we might be interested in what are the other clouds actually? What are the different profiles here of the different cancer regions? So we can select all of them and give them a name. So that's what we did here. So we selected them and gave them cancer 1 to 6. And then we have non-cancer, and then we have the ductal constant C2 that we already know about. So now we have the selection to the left, and then we deselect the ones we know about, so the non-cancer ductal constant C2. And now we ask the software to help us. So we can now say, okay, please let me know we focus now on cluster 1, for example, the blue one down here. What is the most differentially expressed genes or the most upregulated gene in just this cluster compared to all the others? So what's specific about this one? And then we quickly run that analysis in loop, and then you can see, okay, it's CRISP3. But now I'm not sure, like, is this really true maybe? And I really want to see that myself. So what you can do now is you select CRISP3 and look at the gene expression of CRISP3, and really nicely you can see both in the spatial projection up here. It's very nicely and strongly just upregulated in cluster 1, but you can also see that in the violin plot here, that you really have an upregulated of CRISP3. And then you can go down the list and find other genes that might be interesting and really understand this heterogeneity in the tumor. So to talk about what's coming soon. So if you're interested in testing Visium, you've seen that the standard product is the 4 capture area slide. So you can do 4 sections. In order to make it even easier to just take Visium for a test drive, we have released something that we call the gateway slide, which is just 2 capture areas. That everything else is just the same. You get the same resolution and the same experience with a kitted solution and everything else. And this really allows you to lower the threshold, but just giving Visium a try and really see that what I've showed you now, the breast cancer, that was just one section that we ran through. And with just that one section, you can do so much exploration. If you'd rather work with a service provider that runs the assay for you, we have 2 in Europe that we work with. You can see the links here, Imagine and Newcastle. And then also, what I described so far was mostly focused on fresh frozen sections. And obviously, we are aware that clinical workflows and a lot of researchers prefer to work with FFP, or it's just the standard. So later in a very short time, so still in the first half of this year, we will release an FFP compatible product. So that's going to be the same Visium resolution, but compatible with FFP blocks. And all the key features will be the same. It's going to be kitted, whole transcriptome. And just to give an example, here we have a triple positive breast cancer sample. And here we have the hematoxin stain. And then just to give you some idea of the data is, you can see that we get the same kind of clustering. We can also explore the sample, really, both the gene expression, but also the morphology. And just to give you a few examples, we see expression of ESR1, PGR, and HER2 here, but you can really look at any gene that you're interested in the whole transcriptome. So stay tuned for this. We're gonna launch this very soon. So to summarize, the Visium solution is giving you the opportunity to look at either the whole transcriptome, or you can also do targeted panels. It's a highly sensitive assay. You have high spatial resolution. You cover the entire tissue section. It's kitted, ready to use. If you have used any of 10x products, our customers are very happy that we include everything in the kit. So it's a very nice experience. It's a streamlined workflow. We have software, and our support team is great and happy to answer any questions that you might have. We have 3 pillars with 10x. So we have the Chromium platform, which is working on dissociated cells and allows you to do a lot of different measurements on these. We have the Visium platform we just talked about. And then we also in the future will launch an in situ platform that will allow you to go to subcellular resolution with a targeted panel. So then you might have done some discovery on Chromium Visium and can really go into this super high resolution to find specific targets as well. So that's going to come in the future. With that, thank you for your attention, and I'm happy to take any questions.
Thank you, Florian. There's quite a number of questions, but I'm going to try to combine some of them, and I'm asking you to try to be very concise on the answers so that people can have a break.
Yes.
There's 2 things linked here, and what are the challenges for a new user to start with this technology, and what are the costs?
Great. So the challenges, I think that the strength of this is also the challenge, which means you have like a pathologist that might, or a histologist in general, like someone who's used to working with tissues, and then you have someone who's used to working with next generation sequencing. So I think one of the challenge, or one of the things that has to happen, is a collaboration between those. I think that's kind of what has to happen, but that's also a great chance, because now you can use all the knowledge that pathologists have and combine it with like highly complex molecular measurements and really see what can we learn from each other. So I think that's a great thing. About pricing, I actually don't know, but we have a booth, so please stop by our booth, and then you can get more information about the pricing there.
Okay, so Sarantish, do you want to ask your question? Are you there, Sarantish Klamidas?
That would be great. Thank you very much for your talk. It was really great. Actually, I wanted to ask you how you do the extraction of RNA on the situ, actually. How do you perform this in situ extraction from RNA? Thank you.
The extraction is actually capping on. So you have, you section the tissue, you place it on top in the cryostat in this case right now, and then you melt it on top, and then it's very close by. And then you add a permeabilization reagent that creates holes in the cell membrane, and then the RNA can bind on top.
I see. Thank you very much. Thank you.
That's included in the kit. Yeah.
Okay. Leah Powell, you want to ask your question? Leah Powell, are you there?
Hi. I was just wondering, amazing technology. I was just wondering how you guard the patient safety of their data, just because you didn't go into it.
Yeah. So what we offer is a research-only solution right now. So this is really more on the research. So what we provide is the kit, and then we provide the pipeline that has to be set up on a server, and then also the analysis software. So that's really up to the user to work with that. But all that we have also works, like, for example, in if you're not allowed to use an external server or so on. We have a lot of customers that might not want to do that. You can have everything internally. So I think that would be on your side. Because all we do is like that molecular measurement on the tissue, really.
And then there's more than one people asking if you provide a service for this technology.
Yeah, so I had that slide up. We have the certified service provider. So on our, if you pop by our booth, we can give you those links again if we weren't able to capture them, or you can find them on our website. So we have Imagine and then Newcastle University in Europe.
And a last one about, I think you answered that also, but if there is any specific instrumentation required, I think you already mentioned that as well.
So it's not really. So you have the cryostat or microtome, depending on if it's FFP or fresh frozen. Then you do a microscoping, like a microscopy. So you take an image. You might need an automated stage if you don't have that, because it's like quite a large area to image. Then you go into the molecular biology, which means you need PCR machines and like just standard. And then in the end, a next generation sequencer, which you might have in-house, or there's a lot of core facilities out there or service providers that can help you with that.
Then I might just make a final question myself. I mean, this is a fantastic approach. Another one. What are your advantages in comparison to many competitors for the same sort of approach?
Yeah, it's really, I think, as I mentioned before, what we offer is a real discovery solution. So you get like an overview of the entire tissue. Sometimes you have to pre-select some areas, which seems nice because you're like, oh, I want to study this and this. But as soon as you've done it and you sit with your data, you're like, oh, I should have picked this as well. And I think the great thing about Visium is that you just do the measurement on the entire section. And then in data analysis, that's what you do. You select an area and, oh no, I regret that. I do a new selection and I do a new selection, and really on the whole transcriptome. So I think that is really the advantage that no one else can offer. That like you can really discover the tissue without being biased.